everyone, my name is Erika Beltran. It is September 19th, 2019, and I am doing this class for Physiology. Okay, so I'm going to be doing my presentation on the pancreas and liver and their roles in digestive regulation. So the major digestive functions of the pancreas. The pancreas is both an exocrine and endocrine organ, meaning that it has its exocrine functions of secreting certain things, in this case digestive juices, through a gland, and endocrine functions, which is the release of hormones. So our exocrine functions are the release of enzymes and bicarbonate by the ACE in our cells, and I think this is important to think of histologically. And we look at this histological slide of the pancreas, and we can see our ACE in our cells, this dark stained purple, versus this lighter stained circular almost, and this is the pancreatic islet. See how it looks like an island among all of these ACE in our cells. And our pancreatic islet is going to serve our endocrine function of releasing different hormones from the pancreas. Well, we'll first consider the ACE in our function, the exocrine function via our ACE in our cells of the pancreas. This is most relevant to the digestive interplay we're going to be speaking about. And this secretes enzymes and bicarbonate. So bicarbonate secretion, why would we want to secrete bicarbonate? Well, first of all, think, where is the pancreas secreting these enzymes and bicarbonate into? And it is actually going to be going into the duodenum of the small intestine. So we have a meetup between our liver secretions, our pancreatic secretions, and our stomach secretions, as I'll show you in a picture later. And I'll just go to that photo. And so you see here how we have our pancreas secreting, and it's going to secrete through our pancreatic ducts this bicarbonate and these enzymes, our liver secretions, and our stomach all into the duodenum. So we have our bicarbonate secretion and our zymogens. So let's go to this next slide here. What is a zymogen? So a zymogen is a type of enzyme that is secreted in its inactive form, and it needs to be further activated. So an example would be trypsin, which is the active form of trypsinogen, a zymogen. And trypsinogen is released from the pancreas into the duodenum of the small intestine, where it is activated. So how is trypsinogen activated? It is actually activated by some brush border enzymes that are present in the intestinal wall of the small intestine, and one of these being enterokinase. And enterokinase is able to activate trypsinogen into trypsin, which can then activate chymotrypsin and carboxypeptidase. Some examples. So those are some zymogens of the pancreas. We have, though, a lot of pancreatic enzymes that are able to digest all of the major macromolecules here of our food and what we eat. So we have all of these trypsin, chymotrypsin, and carboxypeptidase that can digest proteins into their smaller building blocks. We have pancreatic amylase, similar to salivary amylase in our saliva, that can digest starch and carbohydrates into its building blocks, into maltose and glucose. And we have pancreatic lipase, which can digest our fats, our lipids, into their monoglyceride and fatty acid components. And we also have our pancreatic nuclease, which can digest our DNA and our RNA, and although that is not as common, it is there in order to digest all the major macromolecules that we may consume. So we have here a signal to release enzymes, which basically is saying that our zymogens something needs to change it, something needs to activate it. And our signal to release these enzymes I will actually discuss in the next slide. We have our pH of intestines versus the pH of the stomach, and that is important in considering our role of bicarbonate. So our stomach has this highly acidic chyme due to the release of hydrochloric acid in the stomach, and our stomach is equipped with this thick layer of mucus that helps prevent the stomach from digesting on itself, from digesting its own tissues. But our small intestine, our duodenum, is not equipped for that. So, and the duodenum is just the first part of the small intestine, but speaking of it here, because it is where all of these are secreted. So our bicarbonate is secreted from the pancreas into the duodenum, and it serves as a pH buffer. It is able to neutralize the pH of the acidic chyme, and we can think of it in the same way that it neutralizes the pH of our blood, and that it serves as a buffer in our blood. And we can see a lot of similarities here. We see that it also utilizes carbonic anhydrase, the main enzyme of the pH of the blood buffer. When it works as a buffer in the blood, it uses carbonic anhydrase in the pancreas and duodenum. And it also, we see this chloride shift. So which anion is used when bicarbonate is secreted into the lumen? The cell absorbs chlorine, and this is in order to chloride, and this is in order to maintain homeostasis. It's not necessarily maintaining a neutral net charge, because we know that our membrane potentials in our membrane are just different. We have different types of anions and cations in our intracellular fluid versus our extracellular fluid, but this maintains homeostasis. It maintains a sense of normalcy in those charges. And we have brush border enzymes, as I discussed. Enterokinase is one that'll help to activate trypsinogen into trypsin, but our brush border enzymes also serve an important purpose in further digesting these proteins and starch and fats and DNA and RNA down after these pancreatic enzymes have digested it. It further just gets it into those nice and simple components in order to be absorbed by the small intestine. So important hormones to consider are pancreatic hormones, which is our endocrine function of the pancreas, insulin and glucagon, which act interchangeably or antagonistically on blood glucose levels. And we have our intestinal hormone. CCK is an important one, and it stimulates the gallbladder to release bile, and the pancreatic enzymes released from the pancreas. Secretin helps to stimulate that bicarbonate release from the pancreas as well. So CCK and secretin are some intestinal hormones that can help to begin the digestive process in the small intestine. Before I move on here, some other things that help to signify the digestive processes to begin to take place is the presence of food in the small intestine, the movement of the small intestine, the stretching of the small intestines due to this presence of food, as well as neural signaling, which is too much to talk about in this presentation, but neuronal signaling can also help to begin all of these processes. So the major digestive functions of the liver. I briefly want to talk about the hepatic portal system, as it is not really a digestive function, but it is an important thing to consider because it is a role of the liver that involves the digestive system. Once blood has moved out of all of our gastrointestinal tract, it is pulled through these capillary beds. You can see these capillary beds here and here, and rerouted back into the liver, where the liver is able to detoxify it, remove any of those bad toxins that it may have accumulated. So that is the function of the hepatic portal system. But more importantly for this topic, for digestion, we think about the formation of bile. So the liver actually forms bile. It is not formed in the gallbladder, as many may wrongly assume. The gallbladder simply stores and concentrates the bile. And we can see the gallbladder here, this little green thing right coming off of our liver, and it stores and concentrates the bile. And the gallbladder is actually not important for life. It's not vital for life. You can get your gallbladder removed, and the bile duct here that is secreting bile into the duodenum will just enlarge and serve that role. So the formation of bile, why is bile so important? And because bile is necessary for digestion of fat. And this is the fat emulsification. So bile is made out of 3 components: bile salts, bile pigment, such as bilirubin, and cholesterol. So bile salts are going to be the one component that is recycled, that is going to be reabsorbed by the small intestine and reused. Those bile pigments and cholesterol will actually be excreted into the feces. But our bile salts have these amphipathic qualities that allow for the emulsification of fat, for these large fat droplets to form micelles that increase the surface area, that make it soluble in water, and that allow for that pancreatic lipase to get in there and digest all of those lipids down into their fatty acid and monoglyceride components here, that are able to just freely diffuse across the membrane into the cell. And right here we also see our cholesterol component, that it needs to move via transport proteins, which we had discussed in one of our discussions earlier in this course. So here again, this is just an overview slide. We have our stomach, and we can imagine all of our food coming from our esophagus up here into our stomach, being churned and hydrochloric acid released and starting to be broken down and formed into chyme, a solution called chyme. It is going to be passed through the pyloric sphincter into this duodenum, and is going to mix with the pancreatic juices, the enzymes from the pancreas, the bicarbonate from the pancreas, and bile from the liver. So I want to consider 2 pathologies, and I think that it is important to consider pathologies because we see how vital these functions are to our digestive system. So pancreatitis is actually can be caused by pre-activation of pancreatic enzymes. So it can cause the inflammation of the pancreas because these enzymes are activating in the pancreas before the small intestine, and the pancreas is not equipped for this activation. So it causes inflammation. And if the pancreas is inflamed, what does this mean? This means that the pancreas is not up to par, it is not serving its function as it normally does, so it will not release as many pancreatic enzymes as it usually does. And if we do not have those enzymes released, if we do not have that bicarbonate release, we can actually see malnutrition occur as a side effect of pancreatitis, because those enzymes are not there to break down that proteins, to break down that starch, to break down that fat. And so our stomach, our small intestine, is not able to absorb those macromolecules in their intact form. They need to be broken down in order to be absorbed. So we may be eating something, but if you have pancreatitis, you may not actually be absorbing and digesting those nutrients, so malnutrition can be a side effect. And cirrhosis, which forms this fatty scar tissue, so not fatty tissue, scar tissue, fibrosed tissue of the liver, also impedes liver function, and that means that the liver will not be able to form bile. So what may happen if the liver can't form bile? What are those effects on digestion? We would see it is much harder to absorb fat because bile is such an important component of absorbing fat. So we can see that we may have trouble having fat-soluble vitamins, as those need to be consumed with fat. So we may be lacking our fat-soluble vitamins. We will not be able to absorb fat in the same way as we once were, which can lead to indigestion issues, bloating, possible constipation. So these are just some of the ways in which the pancreas and the liver help to regulate digestive function.